RNA techniques and modifications I have. So we'll look into it one by one. Yeah. So in vitro diagnostics, why do you need molecular diagnosis, especially in infections and many other diseases like genetic diseases where there are mutations? Is basically it is a definitive diagnosis that is sometimes needed, and sometimes you cannot. I mean, especially for infectious organisms, they don't grow in the cultures, and you can't have a definitive diagnosis. So आपको molecular test का जरूरत पड़ती है, right? In vitro diagnostics have played a crucial role in healthcare. These are traditional diagnostic methods like culture, immunoassays, and all those things. They are indirect ways of measurement, sometimes cumbersome, labor intensive, and not always very specific. So PCR started replacing these conventional detecting methods since 1985, and it was put to widespread use during the COVID-19 pandemic. Nucleic acids, 5 versus. Okay. Okay. वो देख लेना ऐसे साइंटिस्ट का है कि नहीं को देखना श्योर चलिए देख मीटिंग पेपर लेता हूँ थैंक यू सॉरी nucleic acids basically DNA RNA different types of nucleic acid based tests are there. We'll be discussing some of them in detail. Not all of them are going to be discussed in detail here, and I'll give you some links where you can have a better understanding of these things. Right. So this is a flowchart showing the historical importance. Conventional PCR invented by Kerry Mullis in first application 1985. Then later on in the late 80s got multiplexed. Then you had something known as nucleic acid based amplification, NASBA, which got widely utilized. Then you had LAMP, RCA, all these things. So this is just for your understanding which are there. And it is a continuously evolving thing, latest being the CRISPR-Cas thing, which you would have heard about. They had a diagnostic test, I think they called it, I mean, especially during COVID, for early detection, early turnaround time, they used some CRISPR-Cas based system. So here, I think, yeah, it's, yeah, here, let's see it now. Sherlock, CRISPR-Cas 13 test. FDA had filed for this in 2020. Right? So why do we need nucleic acids? Because they are direct. They are not an indirect form of measurement. They are very sensitive and specific also. You can detect chromosomal abnormalities, mutations in cancers, some genetic diseases which are there, which are inherited disorders, and antimicrobial drugs. So you can look across the spectrum, like you have things like biofire, where you want to know what is the infecting organism in case of common cold. So across the spectrum, you can multiplex those genes and you can identify them quickly instead of individually trying to identify them. So recent developments in molecular tests have significantly altered the way we look at disease, the way we diagnose them. And broadly, there are two general methods for examining the infectious ones. One is microbial phenotype characterization, where you're looking at the stains, various stains that are there under the microscope, and you're doing culture. Second way is by nucleic acid techniques. So the conventional tools, examining physical characteristics, looking at the protein markers, chromatography, antibiotic susceptibility testing, and so on. Why they are not preferred? Because one is takes time. Lab safety, it has to be done in a separate area. It is not always definitive, and you get limited information. There are indirect methods of looking at these things. Current technologies in molecular diagnosis. This is an important slide which will help you. Just a sec. Yeah. So conventional molecular tests include PCR, which can be further classified as nested PCR, multiplex PCR, reverse transcriptase PCR, quantitative PCR. Then you have Sanger sequencing, and you have newer molecular tests like isothermal amplification methods, which involve LAMP or loop-mediated isothermal amplification, recombinase polymerase amplification, nucleic acid sequence-based amplification. Then you have digital droplet PCR, CRISPR-based assays, and all of this can then be engineered into something as small as a microfluidic platform, giving rise to your point of care devices, right? And next generation sequencing. So we'll look at them one by one. Out of this, the LAMP is something which is very interesting and different from your conventional PCR, because this does not need the extreme temperature. It is isothermal. So it's a kind of isothermal amplification. Unfortunately, the primer over here, they need 6 different primers. You have a forward inner primer, consists of the F2 region at the 3' end, and then F1C region at the 5', which binds there. Then you have backward inner primer. So these details, it may look like there's a lot of thing going on. What happens essentially is it forms a loop with the tablet strand, and it keeps on extending. And again, the 3' and 5', and again they form further loops like hairpins over here, as you can see, and this thing continues. So this is the main advantage is it is quick. You can multiplex it, and you don't need extreme temperature. So you have point of care devices where these things can be seen, right? Yeah. So these are the steps of it. You don't need to go to in detail first time. अगर हमको नहीं भी समझ में आया, it may take some time. You can watch some videos. Basically, this loop, it is the same thing. The amplification starts with the F2 region. It hybridizes with the F2C region of the target DNA, followed by F3 primer. So all these things, there are different primers, different sections forming a loop. And once this F2 is binding here, it is forming another loop like this. So it's going like a S, like a serpentinous uh, form is taken, and it keeps on increasing, right? Next is the RPA, recombinase polymerase amplification. It's another method of isothermal amplification where there are four proteins mainly: recombinase, single strand binding proteins, and strand displacing polymerase. They enable exponential elongation. Exponentially, they amplify the DNA without the need of a thermal cycling step, and they can complete the reaction in the temperature range between 25 to 42 degrees. There are different kits and different ways the amplicons are then detected with the help of various enzymes and hydrolysis through ATP, right? And then finally, the third one, that is the NASPA, nucleic acid sequence-based amplification. It utilizes 3 different kinds of enzymes: reverse transcriptase, RNase H, and T7 RNA polymerase. And again, it does not need high temperature like you need in standard PCR. So continuously produces copies of single-stranded RNA. The primer binding, a primer with the T7 promoter binds to the target RNA. Complementary DNA is then synthesized. RNase H breaks down the original RNA, leaving only the single-stranded complementary DNA. The T7 RNA polymerase then uses this complementary DNA to synthesize thousands of single-stranded RNA copies, which then again re-enter the cycle. Applications: preferred method for detecting single-stranded RNA viruses, retroviruses. Common targets can be HIV, respiratory viruses like influenza, SARS, so on. Other bacterial pathogens like mycoplasma and Legionella, Chlamydophila pneumonia, and so on. So these are newer techniques. Main advantage: you don't need a big setup. It is quicker, and it can be done as a point of care system, right? So basically, if you have to write an answer, you have to know about PCR, you have to know about reverse transcriptase PCR, other modifications like isothermal amplification, next generation sequencing. There's a lot of research which goes on in next generation sequencing, and it is very much different from the Sanger sequencing because it helps you to multiplex and it helps you to dive deeper. I mean, you can do longer reads, you can do deeper sequencing as compared to standard Sanger sequencing. And then CRISPR based diagnostics, it utilizes again a set of enzymes called as Cas13 or Cas12. It is very accurate and it is basically, it snips the DNA and you can edit. It is like a DNA editing tool, and it is also used for rapid detection. So it is not only restricted to infectious diseases. There are it has applications in detecting somatic mutations in cancer like EGFR in lung cancer, BRCA1 in breast cancer, and so on. Detecting various genetic disorders like prenatal screening, identifying inherited diseases like cystic fibrosis, even sickle cell, and so on. So overall, PCR and nucleic acid amplification is probably the most important thing you have to know about. And this technology has changed the way molecular diagnosis is done. There are different, depending on the situation and setup, different hospitals or labs would be using different kinds of PCR. You can have simple thermal cyclers, but you can also have more higher end real-time reverse transcriptase PCRs, which are also able to quantify and even multiplex to a certain extent, where there are different primers, maybe 5 6 different primers can be used, and different regions of the DNA, they are checked and they are amplified, right? So PCR is basically the foundation, and it offers advantage that it is as compared to culture and traditional methods, it is quicker, it is more sensitive, efficient, and it is better. So if you look at these charts here, this is the sensitivity, specificity, cost, and time for detecting microorganisms by culture. The same thing when you look at PCR, sensitivity has improved, specificity is similar, cost is a bit higher, time taken is very less. Whereas sequencing, time is still higher, cost is higher, specificity and sensitivity are higher. And emerging technologies, again, time is very less, whereas sensitivity, specificity are very good. This image gives you different types of PCR that are there. You have conventional PCR, and they have different applications and purposes: diagnostic applications, genetic screening, research, biomolecular sciences, so on. USP: it is simple, quick, and sensitive, but requires knowledge of the target sequence. You should have the exact primers that are needed. For RT-PCR, gene expression analysis, research, quantitative analysis, it is accurate. It can detect an infection even in the early phase, but again requires expensive instruments. Multiplexing in PCR, multiple pathogen can be identified, identifies the co-infections effectively, reduced amplification efficiency due to multiple primers. Nested PCR, increasing sensitivity and specificity. Um, you can have longer templates, amplifies longer reads. Clinical and diagnostic applications are there, but the limitations are time, cost, and it can also amplify. I mean, it has to be very sensitive. The contaminants are also amplified. So this is an important slide which gives you different types of PCR. And if you look at PCR, you basically have 3 steps. You have denaturation, annealing, extension, and then it repeats itself. So the DNA strands need to be separated. So with high temperature around 90 degrees, it separates 90 to 95 degrees. It separates the 2 strands, and then it allows the primers to attach to the respective sequences. The primers then elongate with the help of DNA polymerase. It keeps on adding the base pairs, and then again you have a replication. You have an amplicon. So each cycle it doubles. So one cycle you have from one DNA strand you'll have 2. In the next second cycle you'll have 4, then 8 and 16 and so on. So you have something known as the CT number. So each CT number increase means that is one more double. So CT numbers were used extensively in when SARS was there. You can see that you would have seen them in some of the reports of SARS virus, right? Here are 2 videos. I mean, I don't know if you can see them. This will probably help you understand. I'm sure you have seen these kind of videos for PCR. I think all the molecular things, if you mm, watch it on a video, it's better. Are you able to see this on the screen?
No, sir, only link is visible.
Yeah, sorry. Can you see the video on the screen or no? Video नहीं दिख रहा है, रुको. Let me see what I can do. I'll stop sharing this and share the other window. Wait. Or else you can see it later. It will be there in the presentation. It's not that important. Uh, Let me see. This might just work if you see the entire screen. Audio out. मुझे दे नहीं रहा है. It's not permitting me to share the whole screen. Share, yeah, yeah. Now you can see it.
Yes, sir.
Yeah.
American scientist Henry Mullis invented PCR in 1983. His idea was to use two primers, nucleotides, and DNA polymerase to make unlimited copies of DNA. A primer is a 20 base pair fragment of DNA that pairs with the target DNA. The DNA sequence of the primers is so specific that from a background of 22,000 genes, they can amplify one gene. DNA exists as two strands bound together. So for a primer to access the DNA, the strands first need to be separated by heating at 95 degrees Celsius. As the mixture cools to 60 degrees, the primers bind to opposite strands of the DNA, binding so they flank the target region. DNA polymerase can only copy DNA in one direction, so the strands get made in opposite directions, resulting in overlapping copies. This copying of the DNA by DNA polymerase will double the amount of DNA. The reaction is again heated to 95 degrees to separate the DNA. When cooled, the primers also bind to the new DNA, and the DNA polymerase makes another copy. For each PCR cycle, the amount of DNA doubles, so that 2 copies of DNA can become 2 million copies in just 20 PCR cycles. The flanking placement of the primers limits the length of the amplified DNA. This DNA can be visualized on a gel at the end of the amplification and will be one discrete band. Heating the mixture to 95 degrees destroys DNA polymerase. So fresh DNA polymerase gets added after each heating cycle. This manual addition was very time-consuming and error-prone. But in 1988, Randall Saki from Cetus Corporation used a different DNA polymerase for PCR, one from Thermus aquaticus, or TAT. Thermus aquaticus is a bacteria that lives in hot springs, so it isn't destroyed during the 95-degree heating step. The use of TAC polymerase means that PCR can continue to the end without having to add more proteins. Cetus also created an instrument to automate PCR. The first thermal cycler, the TC1, automated heating and cooling using a metal heat block and inbuilt computer. This automation made the process of PCR far easier. The first commercial real-time PCR instrument was made in 1996 by Applied Biosystems. This instrument monitors fluorescence in the PCR tube as it is happening. DNA production is measured using 2 primers and a probe. A fluorescent dye is attached to one end of the probe and a quenching dye is on the other end. When the probe is intact, the quencher stops the fluorescence. The probe sits close to one of the primers. As the TATA polymerase copies the DNA, it cuts up the probe and releases the dye and quencher. The quencher is no longer near enough to stop the fluorescence, so light gets emitted. The camera then records the fluorescence. The increase in DNA during PCR can be seen on the computer screen in real time as the PCR is cycled. The power of real-time PCR, also known as quantitative PCR or qPCR, is to see when the PCR is doubling each cycle without stopping the reaction. In this doubling phase, the quantification of DNA is possible. In the first few cycles of the qPCR, the amount of fluorescence is below the camera's detection limit. Then, as DNA accumulates, the fluorescence in the tube becomes detectable over the background. At this point, the fluorescence doubles each cycle. This doubling is the exponential growth phase. As millions of copies of DNA accumulate, the reaction slows down to a linear rate and then reaches a plateau. The higher the starting amount of DNA, the earlier the PCR will appear above the background. The amount of DNA in different samples is measured using a threshold line. The point that the fluorescence passes this threshold is the cycle threshold, or CT, of the PCR. The threshold is set above the background and during the exponential phase of the PCR. The CT value is a measure of how much DNA is in the sample. But the CT value can't tell you the exact amount of DNA in the tube. It can only tell you a relative amount. For example, a sample with a CT of 10 will have 2 times more DNA than a sample with a CT of 11. That means that a difference of 2 cycles is 4 times more DNA, and 20 cycles is 1,000,000 times more DNA. So if you want to know the exact copies of DNA in a tube, you need to compare it to a known amount called the standard. Standards covering the full range of qPCR cycles can make a standard curve. Then when you know the CT of a sample, you can read the DNA copies from the standard curve. It's also possible to amplify more than one DNA target in the same tube. Each target uses a probe with a distinct colored dye. Each promo and probe set can target different locations in the DNA or even slight changes in the sequence. This change can be as small as 1 nucleotide in the DNA sequence. PCR and real-time PCR have many uses, for example, in diagnosing disease and in medical research. Top of mind is the detection of viruses and bacteria from respiratory infections. Most of us have had a swab or given a saliva sample for one of these tests. From this sample, PCR is used to identify which viruses or bacteria are present. Different colored probes in one PCR reaction allow for the detection of many targets in one PCR. For example, it's common for one test to detect influenza A, influenza B, and SARS-CoV-2. These are yes-no tests, meaning they report if a virus or bacteria is detected or not. But for COVID-19, the CT value of the real-time PCR is also helpful for contact tracing. A low CT means that the person has a lot of virus present and is more likely to be infectious. Tests over time can tell if the person is early or late in their infection and when they were infectious. The discovery of PCR was a fantastic insight. PCR enables disease diagnosis and a greater understanding of biology. Subscribe to Clever Lab to learn more about human biology.
Yeah, it was a nice video. That's why I thought I should share it. Yeah. So wait, let me again share the screen. Where were we at? Yeah. So, right. So despite the advantages, yeah, the other one was cellular sequencing. You can watch it. I'll share the PPT of the cleaner upside. Same here. I have presentation. तो आपको क्या दिख रहा है? या वेट, ये वो तो कर दिया रे मैं। फुल स्क्रीन दिख रहा है? नहीं दिख रहा है आपको? वेट, आया? या सो या सो लाइक एनी मेथड, जैसे एडवांटेजेस होता है डिसएडवांटेजेस भी है। सो डिसएडवांटेजेस और लिमिटेशंस क्या है? आरएनए अगर आप एम्पलीफाई कर रहे हो, इट नीड्स टू बी टेकन केयर, आई मीन वेरी केयरफुली। इट गेट्स डिग्रेडेड easily. There may be variability in reverse transcription efficiency, dependence on stable I mean the controls and standards has to be very stable. Vulnerability to contamination, errors, अगर sequence में थोड़ा गड़बड़ हुआ तो you will not get what you're looking for. Appropriate controls and misinterpretation of amplification works. Right. So applications are multiple. I mean, कोई भी infectious disease हो, you can detect it. Detection of variations and mutations in genes, inherited disorders, track presence absence of DNA anomalies, prenatal diagnosis, ये सब चीजें आपका यूजफुल है। डीएनए फिंगरप्रिंटिंग और फॉरेंसिक केसेस, वो सब चीजें हैं। Now coming to this thing again, loop mediated isothermal, these are interesting things because like we saw, they operate at a temperature which is far lesser than the temperature needed to denature or separate the two strands. It is very quick. In 30 to 60 minutes, you will get it. Ideal for point of care. And these are very much sought after methods. LAMP is one of the commonly used isothermal amplification techniques. So it has enhanced precision. It can also tolerate, I mean, inhibitors which may be present in various biological matrices, and it is definitely a better method than standard PCR. So the LAMP technique is characterized by 4 to 6 primers. We've already seen all that, and this table basically gives you a description of what those primers are doing and the properties of that. So what are the inner primer, what the inner primer is doing, the forward inner primer, the backward inner primer, and so on. So you don't need to waste too much time on this. So for all the molecular techniques, it is better if you see a video. It gives you a better understanding of the process. I don't. Yeah, sure. I'll definitely do that. Definitely do that. So now this table is probably good if you want to write an answer. It gives you a comparative between the different types of PCR that we saw. The newer isothermal amplification methods. So you have LAMP, RPA, SDA, RCA, SDA, and so on. So, like for example, for LAMP, it has high specificity, it is quick, visual detection is there, inhibitor, it is resistant to various inhibitors in the process. Disadvantages: complex primer designing is required. It may get contaminated due to aerosols and so on. So all these things are different kinds of isothermal amplification methods which are there. You need not remember all of them. Even if you remember one or two as a part of your answer, I think it should be more than enough. Sorry. Then sequencing. So there's a lot which can be talked about over sequencing. We'll have a separate lecture on sequencing. So first generation Sango sequencing was accurate, but it was very slow and high costs were there. Now you have next generation sequencing through Illumina, ABI, then Iron Torrent, Oxford Nanopore. All these things are the newer generation sequencers, which gives us better, much more deeper reads, longer reads, and they do it quickly. Initially, the standard sequencing used to take days for one complete cycle sequence to be completed, whereas nowadays you can do it in a matter of hours. So, in addition to this, there is something known as microarray where you have different wells where you can stick these primers, and if the molecular or the nuclear acids are present, they will attach and hybridize. And on hybridization, it will fluoresce, and you have multiple small wells having different kinds of primers. So it is good for multiplexing. These are available as commercial kits, and they are well standardized. There is an image which I'll show you. Yeah. So it basically needs a cDNA probe, which are then fixed in these wells. They are stuck to these wells. The microarray is prepared, and when the sample is applied, you will get different kinds of fluorescence based on the concentration or presence or absence of those specific sequences in those wells. So it is very good for screening, and this is known as microarray technology. It is a kind of molecular test that is done for screening for different kinds of mutations in the population. Then you have droplet digital PCR, CRISPR-Cas, and something known as APTA sensors. So droplet digital PCR is also a kind of PCR where it is multiplexed by difference in the physical properties. You have oil and water, so they are not visible, and it's a microfluidic chip. There are different small, so like the wells were there in a microarray. In droplet digital, it's a standard slide, but there are different zones where you have oils which are separated by oil and water based compartments. And in these compartments, you have those, where it is where the hybridization take place. And specific timers are attached to these compartments, and you will see you can probably multiplex some 6 or 8 different uh, areas, I mean, mutation sequences of interest. Similarly, this CRISPR-Cas, again, you will need to look at a video because it is difficult to understand from these images. It basically snips the one section of the RNA, and it hybridizes. Basically, there are enzymes called as Cas 12 and 13 are important here. And finally, once the hybridization is complete, the target is achieved, it fluoresces and gives out a light. Similarly, there are optical APTA sensors which are giving out signals and detected by electrochemical signals are there. So these are the novel kinds of molecular techniques which are available. CRISPR is basically one thing. Again, we'll need another session probably, or I can share the videos for CRISPR. It is more than a detecting tool. It is also used as a editing tool. This is, you may have heard of CRISPR, especially during the SARS COVID pandemic. Now coming to microfluidics, these are novel point of care devices where you are not needing big analyzers, and in these small spaces, the sample or the compartments they are made to flow in specific areas. There are primers which are probably embedded in specific sections, and as the um, uh, nuclear material flows or passes through it, it gives it's like a small chamber which is doing a PCR reaction, right? And it is doing isothermal amplification, not a traditional PCR reaction. With the isothermal amplification, it then gives you the signal. So there are quite a few microfluidic devices which are being tried out, especially for infectious diseases. They are helpful to identify infectious organisms quickly. But this is mostly it is still in the research phase. Routine use in diagnostics is there, but not so much. Finally, the aptamers. These are basically single-stranded DNA or RNA nucleotides. They exhibit specific interactions with targets similar to antibodies. Advantages: they are not so costly. They are not easily degraded. They are easy to monitor and detect, and you can reuse these kind of antibody DNA RNA complexes easily. So for future, there is a lot of work which is going on, and which is utilizing LAMP, various isothermal methods, miniaturizing them, putting them in microfluidic chips, and also the amount of data that is coming out is, it needs to be interpreted So there is a role of AI. Obviously, AI is there everywhere, which can help us in developing, optimizing these processes. So to conclude, there are many different nucleic acid-based methods. It is a rapidly evolving field. Point of care devices with the help of microfluidics is a game changer. Multiplexing capabilities have improved, especially with microarray and little droplet PCR. One can detect disease-causing gene mutations, infections, can track drug resistance. All these molecular tests have now become quicker, more accessible even to the common public. Only limitations are there are potential for contamination, and if that happens, those contaminants also get amplified. So this is basically an overview of the role of various nucleic acids in diagnosis of molecular and infectious diseases.